Welcome to our educational webinar titled Modeling Arrhythmias Using hPSC-Derived Cardiomyocytes and Tracking Their Excitability. My name is Coraline Lewis, and I'm a Product Marketing Manager at STEMCELL Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speakers today. Dr. Vincenzo Macri is a Senior Scientist at STEMCELL Technologies, developing products to support cardiomyocyte stem cell research. Dr. Macri completed his PhD in Physiology from the University of British Columbia. He then worked as a postdoctoral research fellow at Massachusetts General Hospital and Harvard Medical School. His areas of study included human genetics, cardiac arrhythmias, cellular electrophysiology, and cardiomyocyte stem cell biology. Dr. Stacie Chvatal is a senior applications scientist at Axiom Biosystems and plays a leading role in developing Axiom's SIPA analysis tool to streamline MEA cardiac analysis. Dr. Chvatal completed her PhD in biomedical engineering from the Georgia Institute of Technology and Emory University. She then continued her research as a postdoctoral fellow at Georgia Tech. Her research was in neurocontrol of movement in healthy and impaired nervous systems of human and animals. Let me now hand you over to Drs. Macri and Chvatal to begin the presentation.
Thank you, Coral. The outline of today's presentation will be covering a few main topics: the overview and generation of cardiomyocytes from human pluripotent stem cells. We'll then move on to how we can use those cardiomyocytes to model cardiac arrhythmias from genetically engineered hPSCs and patient-derived hPSCs. And then finally, we'll go over the MEA or microelectrode array technology as a means to measure excitability and arrhythmias abnormalities with hPSC-derived cardiomyocytes. So to begin with, just want to go over the workflow that we use to make cardiomyocytes from human pluripotent stem cells. So we start with the high-quality hPSCs that are cultured and maintained in our mTeaser medium. We then enter the cardiomyocyte differentiation protocol using the STEMDiff cardiomyocyte system. Here, this set of media system can be used to push the hPSCs down their developmental lineage to cardiomyocytes passing through cardiac mesoderm and finally into cardiomyocytes. And we'll go through this in a little bit more detail. Finally, once we have cardiomyocytes generated, we can transfer those cells and directly measure them using Axiom's CytoView MEA plate and MEA system. Using both of these systems, we can then take those cells and perform disease modeling experiments, which we'll go over, assess toxicology of compounds on cardiomyocytes, specifically looking at excitability, and also doing novel drug discovery. So the STEMDiff cardiomyocyte system is a multi-component system, which basically takes you through three developmental steps using three different media to bring you to a progenitor state to generate then cardiomyocytes in our maintenance media. I just want to spend a little bit of time talking about the prepping of the cells, the hPSC cells at day -2. So these cells can be maintained in either mTeaser 1 or E8. So these are cells that are dissociated as single cells and reseeded, and then allowed to reach confluency about 95% at day 0. So over 48 hours, it's important to allow the cells to proliferate and reach that confluency before entering the differentiation protocol at day 0. This pre-step or phase is standardized and works across many different IPS lines. So I just wanted to highlight that one component before entering the cardiomyocyte system. So once at day 0, we basically go through three media changes, A, B, and C, every 48 hours. And then at day 8, once the cells reach progenitor state, we then switch to maintenance media. And then over the next 7 days, at day 15, you achieve a confluent monolayer of cardiomyocytes. And then the maintenance media can be used to maintain the cells for a month or longer. And we'll show that the reason to use the maintenance media is a lot of the electrophysiology assays in the field are used between day 25 and day 30. And we also have a dissociation kit, which is a streamlined system to take the cardiomyocytes, the monolayer off, and reseed them, for example, into an MEA, and that's a short 15-minute process. And then we also have a support media and a freezing media, which I won't talk about today, really just focusing on the generation of cardiomyocytes and the processing using the dissociation kit. So why did we decide to create this STEM-diff cardiomyocyte system? So some common issues in the field is efficiency of differentiation, and there's a variability in purity and yield. And another issue in the field is the harvesting and replating of cardiomyocytes for downstream assays. So with the first point, our solution was really to come up with a standardized differentiation kit. And the three things that we really focus on are the standardization of the seeding density before entering the differentiation protocol. And this has been optimized and it can be used across a number of different IPS lines and ES lines, which I'll show. We achieve well over 80% cardiac troponin T positive cells, and we have a very high yield of over 1 million cardiomyocytes from a single well of the 12-well plate. With the harvesting and replating, we really wanted to streamline this process and to make it really simple and easy to use. So that's where the association kit comes in. So again, it's a standardized protocol, and you can collect that population of cardiomyocytes and achieve greater than 80% viable cardiomyocytes. So if you do want to replate into an MEA, you can be sure that you'll have a large number of viable cardiomyocytes to use in your measurements. So here's just some images showing how the hPSC cultures change during the step process using the differentiation media. So here is an hPSC culture maintained in aggregates in mTeaser. And here at day 0 is a single cell monolayer of hPSC. So this is how the cells should look like when you add media A. At day 2, after the treatment of media A, be entering mesoderm. So again, you'll have a tightly packed confluent mesoderm cells. At day 4, after the application of media B, at day 4, you'll have cardiac mesoderm. And at day 6, once in media C, you'll enter into cardiac progenitors, and then again another application of media C, and those progenitors start to switch into early cardiomyocytes. And we do see cardiomyocytes as early as day 7 and day 8 in our protocol. And then switching out media C and maintaining it in the maintenance media. And again, we recommend changing it every 2 days. When you reach day 15, you'll have a robust beating monolayer of cardiomyocytes. And here's an example of how those cardiomyocytes look in culture at day 15. So this is a confluent beating monolayer of cardiomyocytes. So you can see that it's intact, it's synchronous, and they're beating along happily in our maintenance media. So what about the performance with respect to using different ES and IPS lines? So here I'm showing you 3 examples. So 1 ES and 2 IPS lines. And here is flow data showing the staining for cardiac troponin T. And in this example here, we're getting well over 85% cardiac troponin T positive cardiomyocytes. So in aggregate, for biological replicates, you can see in the bar graph for percent CTNT, we're getting well over 80% for all 3 hPSC lines. And then next to that is the cardiomyocyte yield. And here we're showing, again, as a biological replicates of 3, we're getting well over 1 million cardiomyocytes. And that's from a single well of a 12-well culture plate. So the total kit together, you can produce well over 50 million cardiomyocytes by using our kit. So this is the morphology, an example of morphology of a single cell cardiomyocyte. So again, this is hPSC's differentiated cardiomyocytes, and then dissociated and replated using our dissociation kit. So you can produce single cells and stain and do confocal imaging. So here's using cardiac troponin T, and here we see the characteristic striations, what you would expect for cardiomyocyte. In brief, I've went over our cardiomyocyte workflow to generate cardiomyocytes from a starting population of high-quality hPSCs maintained in our teaser media. I want to now take you through an example of disease modeling, toxicology screening, and drug discovery using our cardiomyocytes generated with the cardiomyocyte system and integrating it with the cytoview plates and the MEA system from Axion. So the first example I'll be talking about is disease modeling the long QT type 2 syndrome. So just a brief overview of what long QT type 2 is. So if we look at A, the ventricular action potential, in black is a normal action potential profile. So we have the early phase is a rapid upstroke, which is ventricular depolarization. Then we have a plateau phase, and then a repolarization phase, which brings it back to the resting membrane potential of the cardiomyocyte. In long QT, we can see the action potential in the red line. There's not really an effect on the upstroke or the plateau phase of the action potential, but it's really an effect on the repolarization phase of the action potential, such that there's a delay in the repolarization. So here it's denoted as a slowed repolarization. So with long QT, it's specifically type 2, it's specifically due to a defect of the hERG current or the hERG gene. The normal hERG current, which is represented in blue, is an outward going current, and that repolarizes the ventricular action potential back to resting membrane potential so that the amplitude is larger. But in the LQTS situation, the hERG current is much smaller. And so that results in a delay in the repolarization that you see in the action potential. So how does that relate to a human ECG? So in a surface electrocardiogram, here briefly go over the P wave, which denotes atrial depolarization. QRS is ventricular depolarization, and the T wave is ventricular repolarization. So we're really interested in the QT. So the time points between ventricular depolarization and the T wave, which is ventricular repolarization. And again, if we follow the dotted lines between the black and the red, which is the normal, and the one where there's a defect in the hERG channel, we can see that there is a delay in the T wave in the red line. So this is long QT type 2 syndrome, specifically caused by a defect in the hERG current. So why is this important? So I'm showing you one cycle here of the cardiac cycle. But if this goes over time, for many cycles, eventually there is a potential for it to go into an arrhythmia. So here is an ECG strip showing a normal rhythm. Then we can see there's a long pause, and then the ECG then moves into Torsades-Dupont phenomenon, where there's an unstable excitability rhythm, and that could be fatal. So this is why long QT syndrome is important to understand and to modify. So specifically, I'll be talking about the hERG channel and how we have modified using CRISPR-Cas9 the N-terminal portion. So here is a schematic of the hERG ion channel. And so here we're focusing on the N-terminus region. And I'll then be talking about a patient with an inherited mutation in hERG that's located in the C-terminus. And so what's interesting about these two point mutants is that they affect different parts of the channel, but they have the same net effect, which is delayed repolarization. So just to briefly go over how we generated our CRISPR-Cas9 edited, hERG edited lines. So we used CRISPR-Cas9 along with cloner, which is our cloning media. We use cloner because it increases the efficiency of generating colonies. So we can generate, in this example, 30% clones getting back when you single cell seed them. And then what we can do from then is then pick those colonies. In this example, we picked 60 colonies and then cultured them after using cloner in mTeaser, and you can see that we're getting nice aggregates back. So we took those clones and then had them sequenced, and what we found was in clone 2 here, one of the clones, we see a deletion in one of the alleles, where in the second allele there isn't this 4 nucleotide deletion. So here we were able to generate a heterozygote deletion in one of the alleles of the hERG channel where the non-transfected control had no effect. And we also looked at off-target effects for 3 most likely ones, and we found no off-target effects. So again, looking at the hERG channel here, so this CRISPR-Cas9 really affected the N-terminal region. And in the wild-type allele, you see the N-terminus is quite long. However, in the mutated allele, that 4 nucleotide deletion causes a shift here, and it causes an early stop codon. So basically making this allele null, resulting in a heterozygote mutation of the hERG channel. So what we end up doing is then taking those, the control and the hERG edited line, and then set them up in our cardiomyocyte differentiation system. So again, using our single cell seeding at day -2, allowing those cells to grow to confluency, and then entering the differentiation workflow. At day 15, we achieved monolayers of cardiomyocytes from the control and hERG edited line. We then took those cells at day 15 from the control and edited lines, harvest them with a dissociation kit, and then replated them on the MEA, and then maintain those cells with our maintenance kit for another 10 days. And we did our electrophysiology assays at day 28. And here's an example of the site of view plate. So here we can see the electrodes from the site of view plate in the well. And here we can see that we're basically recovering the monolayer of cardiomyocytes. So just a brief overview of the MEA with how it relates to the underlying currents that make up the cardiac action potential, which we just covered in a few slides back. So here are the underlying ionic currents that represents the depolarization phase, so the sodium current, the plateau phase, which is the L-type calcium, and then really we're focusing on this phase 3, which is the repolarization phase, which is made up of really the hERG current and KCNQ1. And so with the MEA, what we're measuring is a field potential, and the field potential has a spike amplitude, and that spike amplitude relates to the depolarization of the exponential. Then there's a field potential duration, which is equivalent to the repolarization phase. And then we can also measure beat period, so between the cycles. And specifically now, we're going to be looking at the edited hERG mutation, which causes the heterozygote hERG in that edited line. And so we'll be expecting an effect on the field potential duration, since this is representative of repolarization. So here is an example. The black line is the control, and the red line is the hERG edited. And here we can see immediately two phenomena, is that with respect to the control in black, we can see the FPD phase is prolonged in the hERG edited line, and then also the next cycle is also prolonged. The spike amplitude is reduced, which is expected as well in this hERG edited line. And so if we then now look at not just 2 cycles, but over 5 cycles, we can see that the delay is present and that there's a delay also in the cycling. And then we talked about the prolongation of the repolarization phase, but really about the Torsades-DuPont and this unstable excitability profile on the ECG strip. So can we see that in culture? And so here is now a 50-second recording using MEA on the control. We can see that the beat cycle is quite consistent and regular. However, with our hERG edited heterozygote line, we can see that there's a delay, and then that over time there's an unstable phase, and then it spontaneously goes into a rapid beating, similar to what you would see in Torsades-DuPont. So here in this example, we were able to generate a heterozygote mutation using CRISPR-Cas9 and cloner to then take those cells and then generate cardiomyocytes and model long QT type 2 in cardiomyocytes. So now I wanted to switch to a hERG point mutation that has been inherited through a generation, in a family. So this is IPS lines that we had acquired from the Kelly Fraser's lab at the University of California in San Diego. And this paper has been published recently. And what they found is a point mutation in the C-terminal region of the hERG channel located here. And that also caused an early stop here. And what they found was that affected individuals with this 3003 G to A point mutation were also heterozygotes, and so one allele wasn't expressing as much as the other compared to the non-affecteds. So that we went ahead and then acquired an IPS line of the non-affected grandmother, so 2-6 here, and then the affected granddaughter, 2-1 here. So these are IPS lines that were not generated by us. They were generated by the Fraser lab and brought to STEMCELL for us to characterize and make cardiomyocytes using our cardiomyocyte system. So again, we took both 2-6 and 2-1. We made cardiomyocytes from them. We dissociated them and replayed them into MEAs, and this is an example of here in the side of view. Again, bringing back that monolayer, and again, then doing an electrophysiology assay at day 28. And here we can see again the black is the control, so the grandmother, the non-affected, 2-6, showing regular beat cycle, beat period, and shorter FPD. And then red is the granddaughter, the 2-1 affected. And here we can see just off the first cycle alone that the FPD is prolonged, but also that the cycling between the beat periods is delayed and unstable as well compared to the grandmother. Brief overview of what we've done so far. So we've talked about the cardiomyocyte system and Axion's side of view plates and MEA system, and use disease modeling as an example of how we can study arrhythmias in culture. Now I want to focus on toxicology, screening, and drug discovery using both systems. So we can use the cardiomyocyte system and the Axion system to do medium to high throughput toxicology screen. So what we ended up doing is using a 48 well plate and directly seeding our 4 different unique hPSC lines directly into the side of view plate. So we took our aggregate cultures, replated them at day -2 directly into the MEA plate, and then differentiated them going through all these steps we did before, and then basically tracked excitability starting at day 14 moving forward, and basically measuring how does excitability change and how does it normalize, and really looking at spike amplitude, field potential duration, and beat period. And so here are the 4 unique hPSC lines, 2 ES and 2 IPS. And this is really a time course study looking at beat period. And we can see here that there's detectable beat period starting at day 14, and that it's quite stable about day 20 moving onward. And they're pretty much the same across the 4 different hPSC lines. The spike amplitude had a similar phenomenon where it was detectable at day 14 and again stabilized at day 20 and onward. So it was quite consistent. And then also looking at the FPD was detectable at day 14 and really started to stabilize around day 20 across all 4 lines. And so what we ended up doing next then was looking at a hERG blocker. So we've talked about hERG so far, so maintain that theme. We looked at a blocker of hERG, and specifically we looked at E4031. And E4031 is a specific blocker of hERG, and again we'd expect a delay in the FPD since it's affecting the repolarization phase of the cardiac action potential. So again, using 10 nanomolar E4031 after 10 minutes of application, we see a delay in the FPD, and that's an example above. And here is the aggregate numbers across the 4 different PSC lines. So we had a similar effect across all 4, that we're getting about 40% prolongation of the FPD. And with that, that concludes my portion, and I will now switch over to my colleague, Dr. Chvatal, who will explain and go over the Axion MA system and analysis.
Great, thank you. So you just saw some nice data illustrating the development of cardiomyocyte beating using the Derived Cardiomyocyte system. And Enzo showed some nice differences in activity and control in targeted cells as well as the disease line. So now I will show you how easy it is to generate that data using Axion Biosystems Maestro and the Axis software. The Maestro is a multi-well microelectrode array platform with 768 electrodes. We offer multiple plate types and formats, including 12, 48, and 96 well plates. The plates used here in these studies were cytoview plates with 48 wells and a transparent bottom. You culture cells in the MEA plates just as you would any other plate type, and since the Maestro is non-invasive and label-free, you can monitor electrical activity over days, weeks, or even months. The Maestro's comprehensive software, called Axis, makes data acquisition and analysis simple, making it straightforward to monitor cell excitability, track development, and investigate arrhythmia. Here's a video showing the Axis software, which is used for both data acquisition and analysis. The data that you'll see here is data from the study that Enzo showed, tracking the development of beating over time. In the continuous plots, you see the cardiomyocyte spontaneous beating in real time. Each rectangle shows the voltage data recorded on an electrode, and here you see all of the electrodes in one well. The Axis software automatically detects the cardiac beats and plots them in the cardiac beat plots window. You see individual beats detected on each electrode, as well as real-time beat propagation maps and plots showing beat stability, which are important for assessing arrhythmia. The plate-wide activity map in Axis shows the cardiomyocyte activity across the entire plate at the same time. This makes it easy to compare different conditions on your plate at a glance. Axis computes a number of cardiac endpoints from the field potential that you can export. Metrics related to depolarization include beat period, beat irregularity, which is indicative of arrhythmia, spike amplitude, and spike slope. Metrics related to repolarization include field potential duration, or FPD, which again is the time between the depolarization spike and the repolarization bump, or T wave. And Axis also computes FPD coefficient of variation. Since there are multiple electrodes in each well, we can track the propagation of the beat through the network. In this example, the beat starts in the lower right and moves toward the upper left. Axis computes a number of endpoints to quantify that propagation, including conduction velocity, the starting and ending locations of each beat, the number of propagation patterns observed in the well, and the percentage of beats that follow the most common propagation pattern. It's simple and straightforward to analyze data in Axis. To analyze a recording, double-click to enter a plate map if you didn't do this when you first recorded the data. You can also copy and paste plate maps to and from Excel. Right-click to add a cardiac analysis configuration. That's really all you need to do. Axion has optimized these settings for cardiac applications. If you would like more control over how exactly the beat detection is performed, you can double-click on the cardiac beat detector to change the detection settings. Then just push play to look at the data, or push record to save the analysis file. The cardiac beat plots window gives lots of information about cardiac beating in real time. There are plots that show the detected beats on each electrode and markers that show where the FPD was detected on each beat, as well as the spread in FPD across beats. The conduction plot shows beat propagation, and the beat periods and Poincaré plots provide real-time information about beat stability, which is important for assessing arrhythmia. Some beat statistics, like beat period and FPD, are displayed here in the lower right, and they're provided in real time for each detected beat. All of these images are easily copied with a single click to paste into documents or presentations. Axis makes it easy to compare different conditions on your plate by looking at the plate-wide activity map. This activity map shows the spike amplitude across all 48 wells on the plate. Bright colors indicate stronger amplitudes. This was the data Enzo showed where he tracked the development of beating in 4 stem cell lines at once. You can see here that H9 has the strongest beating, and the other lines are beating well also in most of the wells with varying amplitudes. In addition to looking at spike amplitude on an activity map, you can also look at an activity map comparing the beat rate across the plate. So this activity map is a great way to immediately see the activity across your whole plate, giving you an at-a-glance comparison of your different conditions in real time. The analysis file that you save from Axis is a CSV file which has all of the cardiac endpoints, such as beat period, FPD, depolarization spike amplitude, and conduction velocity, as well as others. These endpoints are reported for each beat, and they're also averaged across beats and replicates to report average endpoints for each electrode, well, and treatment group. Axis also has a batch processing option, which allows you to analyze multiple files at once. From here, you can review the endpoints in Excel or use a complementary Axion software tool called the Axis Metric Plotting Tool to load in one or more of these CSV files and make comparisons. This tool lets you compare cardiac endpoints across treatments, time points, and dosing conditions. Use this section of the tool to select files to compare, change display names and file order if you like, and select a new plate map if needed. This section at the bottom has all the plotting options. You can compare across time points or dosing conditions by showing either one treatment per plot or one time point per plot. You can also change the order of treatments in the plot. You can display either standard deviation or standard error of the mean for the error bars, display raw values or percent change from baseline, and even exclude wells that don't meet a minimum activity criterion. With a simple drop-down, you can explore all of the endpoints provided by Axis. For example, here we're looking at how the beat period developed or changed over time for these 4 different cell lines. We can easily look at other endpoints also, such as beat period irregularity, field potential duration, and spike amplitude, for example. You can export the figure for publication or the source data for further analysis, such as testing for statistical significance. Another tool Axion has developed to complement Axis is the SIPA analysis tool. The SIPA initiative, or Comprehensive In Vitro Pro-Arrhythmia Assay, aims to improve preclinical cardiac safety testing. A key part of SIPA is using stem cell-derived cardiomyocytes and MEAs for whole cell assessments of compound safety. To this end, we developed the SIPA analysis tool for even more precise FPD tracking and arrhythmia inspection, and now it has become a powerful tool for all cardiac analysis. To use the tool, a golden channel is selected from all electrodes in the well as one which has a clear, trackable repolarization feature across dosing conditions. Use the section on the right to modify the automatic FPD if needed. The SIPA analysis tool exports figures and spreadsheets containing results. For example, this figure shows the baseline beat waveform in black and the beat waveform after dosing with 10 nanomolar E4031, a hERG blocker like Enzo showed, in red. We can see that E4031 increased the FPD. The SIPA analysis tool also generates figures like you see here on the right, which shows the percent change in FPD after dosing with E4031 at different concentrations. Higher doses caused a greater prolongation in FPD. The inspection window in the SIPA analysis tool provides a closer look at arrhythmia. This data is from the hERG edited cells that Enzo showed earlier. Each circle indicates the beat period for each beat displayed over time. We can see that the beating is very erratic here, with beat periods ranging from 1 to 4 seconds. These plots on the bottom display segments of raw data, and we can see here the unstable beating in these plots also, with some long beats followed by shorter beats, illustrating the arrhythmia. The SIPA analysis tool can also be used for EAD classification. This is a different data set in which the compound caused these early after depolarizations, which we see in the raw data, and also which cause this alternating pattern of beat periods that we see above. You can use the checkboxes here to classify these EADs according to the SIPA categories shown here, and then those labels are included in the output spreadsheet with the data. So in summary, the AXIS software really makes cardiac data analysis simple. AXIS is a powerful software platform used for both acquisition and analysis, and it's easy to use, allowing you to get to answers quickly. Also, there are optional supporting tools, the AXIS metric plotting tool and the SIPA analysis tool, which you can use for data visualization and review. The Maestro multi-well MEA system is a powerful platform which allows you to monitor cardiomyocyte activity over hours, weeks, or months. Using the Maestro, AXIS, and associated software tools, you can quickly compare conditions, monitor cell development, and evaluate arrhythmia. The STEMDiff cardiomyocyte differentiation and maintenance kits, combined with the Maestro MEA system, really enable medium to high throughput drug discovery and toxicology screening in a label-free manner. So now I'll turn it back over to Dr. Macri, who will summarize how these two technologies are really bringing clinical phenotypes to an in vitro model and where we'll go from here.
Thank you, Stacie. So as an overview of what we've spoken about today, we've really kind of highlighted the use of human pluripotent stem cells as a means to make cardiomyocytes using the cardiomyocyte system in a streamlined, easy fashion so that you can generate cardiomyocytes and then using the Maestro MEA system inside of UPlate to really answer questions, as Stacie nicely highlighted, using their platform and software to really streamline experiments so that you can get the answers that you're interested in. For example, the disease modeling that we talked about today, toxicology, and drug discovery. So we hope that we're able to convey two technologies that streamline research efforts such that the pluripotent stem cell technology can be used to make cardiomyocytes and that phenotyping can be made easy and quicker. So I would like to thank all the attendees for joining and listening to this webinar. Both Stacie and I were very happy to give it. And we just want to acknowledge our groups at STEMCELL Technologies and Axion Biosystems. We have quite a robust and large R&D group and a product management group that support our efforts to develop these technologies. Thank you, everyone, for joining.
Great, thanks very much, Enzo and Stacie. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. Let's start with our first question. Enzo, how comparable was differentiation efficiency between control and the hERG-edited cell lines that you used?
Yeah, that's a good question. So they were very comparable. So we were able to achieve well over 80% cardiomyocytes, and we were able to then take those population cardiomyocytes and reseed them to reestablish the monolayer in the MEA plate. So the efficiency between both the edited and the control or the non-edited lines were very similar.
Thank you. Other than using MEA, what are other assays that can be used with these cardiomyocytes for drug screening or toxicity assays?
So here we highlighted the MEA system, as Stacie highlighted with the SIPA initiative as a means to do more medium to high throughput ways of assessing toxicology and drug screening. There are some lower throughput methods that you could do that require single cell. So here's a reconstitution of a monolayer, looking at monolayers of cardiomyocytes. So one can do single cell electrophysiology using these cardiomyocytes. We haven't shown it here, but you could do patch clamp electrophysiology to look at single cell action potentials, or you could use optical mapping techniques. You can look at calcium imaging as well to look at how an individual cardiomyocyte looks like. And our cardiomyocytes, we don't show it today at this talk because of time, but our cardiomyocytes are adaptable to single cell phenotyping so that you could generate the cardiomyocytes as we showed. Using the STEMDiff cardiomyocyte association kit, you can generate single cells. So just you can use a more dilute volume of cells and replate them down to do single cell work. So those are your other options in addition to the MEA.
Great, thanks, Enzo. Another question we have here is that while you're talking about cardiomyocytes in general, do you have any idea what are the percentages of atrial versus ventricular cardiomyocytes or versus SAN as well?
So in these cultures, other protocols and protocols in literature, these ones are predominantly of the working myocardium. So there are going to be majority of ventricular cardiomyocytes with atrial, not as many as SA node or AV node conduction tissue type. So the majority are going to be working myocardium.
Great, thank you. Have you tried modeling any other arrhythmias, such as Brugada syndrome, other LQTS, or CPVT syndromes?
Another good question. We haven't done that. Those are all really good things to try. We hope that our cardiomyocytes will be used in those other systems. We went with HERG as a model for editing, and we were able to get access to the HERG edited patient lines, but in the future I would foresee doing other arrhythmias as well as the ones that were mentioned.
There was mention that the spike amplitude can give an idea of strength of beating. Is there actually a way to determine the force of beating of cardiomyocytes?
In the platform that we've showed here in the MEA, that's really an excitability measurement. So force of contraction, you'd have to use a different platform. The spike amplitude, I'm not sure would be a direct correlate of strength of beating. You'd have to look at force of contraction using either time lapse or looking at the phase difference in the single cell cardiomyocyte.
Great, thanks. I think this is a great question for Stacie. You talked about the 48 well MEA plates. Are there other formats of plates available for use with the Axion system?
Yes, there are. We have also 12 well plates and 96 well plates. So each plate has 768 electrodes total, and then those electrodes are divided amongst the wells accordingly. So for example, in a 12 well plate, you'd have 64 electrodes in each well, or the 48 well plates that we used here and showed have 16 electrodes in each well, while the 96 well plates have 8 electrodes in each well.
Great, thanks very much, Stacie. All right, we have time for one last question. This question is probably for Enzo. How did you get 80 to 90% of cardiac troponin T positive cells?
The means of doing it was really looking at a way of optimizing the protocol. So as I talked about earlier about the entry of the PSCs, so how to actually make the PSCs, so single celling them and then getting them to a confluency over that 48 time period. We spent a lot of time troubleshooting that because some protocols, they have a range for basically troubleshooting the cell density at the start. And so we've spent a lot of time normalizing that process. And we've shown that using that setup protocol for the PSCs before entering the cardiomyocyte workflow, we standardized it and it works across different, so there's no troubleshooting involved. And that really was a help. And then we really spent time optimizing reagents such that the performance would be consistent so that you would reach numbers of 80 and 90% CNT positive cells across a number of IPS lines. So following our simple protocol and really following it, what we found is that there is that robustness of getting consistency across a number of IPS lines. So that's essentially how we did it.
Great, thanks very much, Enzo. Well, okay, I think that's all the time we have for today. I really want to thank everyone for attending, and I'd especially like to thank you, Enzo and Stacie, for your time and an excellent presentation. Thanks again, everyone, and goodbye and have a great day.